You're watching Fictional Scientist, a journey into alternate realities, fractured timelines, and high-stakes thought experiments. 75,000 years ago, something almost killed every human on Earth. We survived, barely. 8,000 individuals left out of millions. For decades, scientists blamed a volcanic eruption, a climate catastrophe. They were wrong. The ice cores tell a different story. Preserved in ancient glaciers, researchers found microscopic evidence of a pathogen unlike anything in modern biology. It targeted one thing: human memory. It spread through water, through breath, through touch, and it was waking up. The first cases appeared in a Norwegian town where glacial meltwater fed the reservoir. People started losing time, losing themselves. When the outbreak reached Washington University for analysis, the lead researcher made a discovery that terrified her. This wasn't a virus that evolved naturally. Someone made this, someone who wanted to erase humanity. And they almost succeeded once before. Dr. Catherine Mills had spent 15 years studying ice cores. Most people found the work tedious, staring at cylinders of frozen water extracted from glaciers, analyzing tiny air bubbles trapped millennia ago. But Catherine saw them as time capsules, each layer telling a story about Earth's past climate, ancient atmospheres, long-dead ecosystems. She had never expected to find evidence of extinction. The cores came from the Greenland ice sheet, one of the oldest continuous ice formations on the planet. Her team had drilled down through layers representing thousands of years, extracting samples that would help reconstruct climate patterns from the last ice age. But one section was different. The ice from roughly 75,000 years ago showed unusual discoloration, faint amber streaks running through otherwise pristine frozen water. At first, Catherine thought it might be volcanic ash from the Toba eruption, a catastrophic event known to have occurred around that time. Preliminary analysis ruled that out. The discoloration wasn't mineral, it was organic. Under high magnification, Catherine observed cellular structures, intact, suspended in the ice like insects in amber. The structures were approximately 100 nanometers in diameter, putting them in the size range of large viruses. But their morphology was wrong. They had an icosahedral protein shell like many viruses, but with additional external structures that resembled the attachment proteins of bacteriophages. She extracted genetic material using standard protocols for ancient DNA recovery. The yield was surprisingly high. Whatever these organisms were, the ice had preserved them remarkably well. The sequencing revealed a genome of staggering complexity. Most viruses have genomes ranging from a few thousand to a few 100,000 base pairs. This organism had over 2 million base pairs of genetic information, larger than many bacteria, and the sequences were unlike anything in existing databases. Catherine ran comparative analyses against every known pathogen: influenza, coronaviruses, herpes viruses, retroviruses, bacteriophages. Nothing matched. The genetic architecture was unique, with coding regions that resembled multiple different viral families spliced together in ways that didn't occur naturally. There were genes for proteins that could modify host cell DNA, sequences similar to CRISPR systems that bacteria use for adaptive immunity. Regions that appeared designed for crossing biological barriers like cell membranes and even the blood-brain barrier. The more Catherine analyzed the genome, the more disturbed she became. This level of sophistication suggested design, but that was impossible. Nothing 75,000 years ago should have had the knowledge to engineer biology at this level. She published her initial findings cautiously, framing them as unidentified biological material of unknown origin. The paper appeared in a specialized glaciology journal with limited readership. A few colleagues reached out with questions. Most dismissed it as contamination. Catherine knew they were wrong, but without more evidence, she couldn't prove it. Then the glaciers started melting faster. The town of Longyearbyen sits on the Norwegian archipelago of Svalbard, roughly 1,200 kilometers from the North Pole. population just over 2,000. The settlement exists primarily to support coal mining operations and Arctic research stations. In early spring, residents began noticing changes in their water supply. The usual mineral taste had vanished, replaced by something cleaner, almost sweet. The local utility company traced it to dramatically increased glacial melt feeding the reservoir system. No one thought it was concerning, just more water from melting ice. Then people started having trouble sleeping. It began with scattered complaints to the town's small medical clinic. Patients described persistent insomnia that defied normal patterns. They would lie in bed feeling exhausted but unable to transition into sleep. When they finally did sleep, usually from sheer exhaustion after days of wakefulness, they woke feeling worse, disoriented, and foggy. Dr. Anders Holt, the clinic's primary physician, initially attributed it to seasonal factors. The Arctic spring meant rapidly changing light conditions. Circadian rhythm disruption was common. He prescribed melatonin supplements and light therapy. Neither worked. Within 2 weeks, the cases multiplied exponentially. Dozens of people reporting identical symptoms. The insomnia was progressing beyond normal sleep disorders. People were going 4, 5, 6 days without any sleep at all. But something else was emerging that Anders found more disturbing. Patients were losing memories, not in the gradual way associated with aging or dementia. This was acute, sudden, selective. A patient would come in for a follow-up appointment and not remember the previous visit. Recent memories vanished first. What happened today? Yesterday? Last week? But older memories remained intact. Patients could describe their childhoods in detail, but couldn't tell you what they had for breakfast. It was as if someone was erasing their memories backward through time. Anders called for emergency assistance from the mainland. Within days, a team of specialists arrived. Neurologists, psychiatrists, infectious disease specialists. They ran every test available. MRI scans showed unusual activity in the hippocampus, the brain region critical for forming new memories. PET scans revealed inflammation in neural tissue. Blood work indicated elevated levels of immune system proteins associated with brain infection. But no one could identify a causative agent. The breakthrough came when Dr. Ingrid Larsen, a visiting microbiologist, decided to test the water supply. She collected samples from the reservoir, from household taps, from the glacial streams feeding the system. Under microscopic examination, she found them. Particles suspended in the water, roughly 100 nanometers. Protein shells containing genetic material. Their structure was wrong. Too complex, too organized. Ingrid ran genetic sequencing. The results came back within hours, and she immediately understood the severity of what she was looking at. This was no ordinary pathogen. The Norwegian government implemented emergency protocols within hours of receiving Ingrid's report. Longyearbyen was declared a quarantine zone. Military transport helicopters arrived carrying biohazard teams in full protective equipment. Every resident was tested. Blood draws, nasal swabs, spinal taps to check for pathogen presence in cerebrospinal fluid. The results were devastating. Over 90% of the population showed signs of infection. The pathogen was present in their bloodstreams, but more critically, it had crossed the blood-brain barrier. Microscopic examination of spinal fluid revealed viral particles actively replicating in the central nervous system. The blood-brain barrier is one of evolution's most sophisticated defenses. Specialized cells lining blood vessels in the brain create a selective barrier that prevents most pathogens from entering neural tissue. But this pathogen had mechanisms specifically designed to breach that barrier. Its protein shell contained surface proteins that mimicked molecules naturally transported across the blood-brain barrier. It was using the brain's own transport systems as a Trojan horse. Once inside the brain, it targeted neurons with terrifying precision. The pathogen's surface proteins had binding sites specifically designed to attach to receptors found on hippocampal neurons. The hippocampus is the brain region responsible for consolidating short-term memories into long-term storage. When the pathogen bound to these neurons, it injected its genetic material directly into the cell nucleus. Inside the nucleus, the pathogen's genes began integrating into the neuron's own DNA. It inserted itself into specific regions of the genome associated with synaptic plasticity, the ability of neurons to form and maintain the connections that store memories. Once integrated, the pathogen began producing proteins that interfered with normal synaptic function. Synapses are the connection points between neurons. When you form a memory, specific neurons fire together, and the synapses connecting them strengthen through long-term potentiation. The stronger the synapse, the more permanent the memory. The pathogen's proteins disrupted this process. They interfered with neurotransmitter release, blocked receptor activation, prevented the molecular changes that strengthened synaptic connections. New memories couldn't form. And in existing memories, the ones stored in synaptic connections throughout the brain, began weakening as the pathogen spread. But the terror wasn't just in Longyearbyen. Glacial meltwater doesn't respect borders. The streams feeding Longyearbyen's reservoir connected to larger river systems. Those rivers flowed south through mainland Norway, eventually reaching the sea. Water treatment plants were not designed to filter out particles as small as viruses. Standard filtration removes bacteria and larger parasites. Chemical treatment kills most pathogens, but this organism was resilient. Its protein shell protected it from chlorine and other standard disinfectants. Within days of Longyearbyen's quarantine, cases appeared in Tromsø, then Trondheim, then Oslo. The symptoms were identical: insomnia, progressive memory loss, behavioral changes. Sweden reported cases in communities along rivers originating in Norwegian mountains. Finland saw infections in coastal towns. Denmark identified patients who had no connection to Norway except drinking water sourced from rivers flowing south from Arctic regions. The World Health Organization convened emergency meetings. Epidemiologists mapped the spread using case reports and water system data. The picture that emerged was terrifying. The pathogen wasn't just spreading through Scandinavia, it had entered the Arctic Ocean through river discharge. Ocean currents were carrying it south along the Norwegian coast, into the North Atlantic, toward shipping lanes connecting every major port. Coastal communities began reporting cases. Port cities, any population center that drew water from sources potentially contaminated by Arctic runoff. The infection rate climbed exponentially. Thousands became tens of thousands, then hundreds of thousands. Samples were being collected from outbreak zones and shipped to research facilities around the world. One of those samples arrived at Washington University in a laboratory run by Dr. Catherine Mills. Catherine was reviewing grant proposals when her assistant knocked urgently. A package had arrived from the Norwegian Institute of Public Health, biological samples, marked urgent and flagged for immediate analysis. She opened the secure container in her biosafety cabinet. Inside were vials of preserved viral material extracted from infected patients. Catherine prepared samples for electron microscopy first. She needed to see the physical structure before genetic analysis. Under the microscope, she recognized them immediately. The morphology was identical to the organisms she had found in the Greenland ice cores 2 years earlier. The icosahedral protein shell, the unusual external structures, the size and proportions. This wasn't a new outbreak. This was the ancient pathogen she had discovered. Somehow, it had been released from the ice and was now loose in the modern world. Catherine assembled an emergency team: virologists, neuroscientists, molecular biologists. The team began comprehensive analysis, while Catherine focused on the genetic sequences. The genome was even more complex than her initial analysis had suggested. Over 2 million base pairs of densely packed genetic information. For comparison, influenza has about 13,000 base pairs. The genome contained distinct modules, one coded for proteins designed to cross the blood-brain barrier, another contained genes for targeting hippocampal neurons specifically. A third module appeared designed for immune evasion. Each module was sophisticated enough to be a complete virus on its own. Together, they formed a weapon of terrifying precision. Catherine focused on understanding how the pathogen disrupted memory formation. She cultured human neurons in the lab and infected them with the pathogen, then monitored what happened at the molecular level. The process was systematic and methodical. First, the pathogen attached to NMDA receptors on the neuron's surface. These receptors play a crucial role in synaptic plasticity and memory formation. By binding to them, the pathogen hijacked the neuron's own machinery for learning and memory. Once attached, the pathogen injected its genetic material through a specialized protein complex. The viral DNA entered the nucleus and began integrating into the host genome. The integration points weren't random. The pathogen targeted specific genes involved in synaptic function. Genes that code for proteins like c-Rib, which regulates transcription of other genes essential for long-term memory storage. Genes that produce BDNF, a growth factor that strengthens synaptic connections. By inserting itself into these genes, the pathogen disrupted their normal function. Neurons could no longer produce the proteins needed to maintain strong synapses. Existing connections began weakening. New ones couldn't form properly. But the pathogen did something else even more insidious. It produced proteins that actively degraded existing synaptic connections. These synaptic pruning factors normally play a role in development. But the pathogen's versions were hyperactive, targeting synapses indiscriminately. The result was systematic erasure of memory at the cellular level. Catherine watched this process unfold in her cultured neurons over days. The elaborate networks of connections between cells dissolved. The neurons themselves remained alive, but the patterns that stored information were being systematically destroyed. It was like watching someone erase a hard drive, one file at a time. She tried various interventions, compounds that could block receptor binding, molecules that might prevent DNA integration, drugs that could inhibit the synaptic pruning proteins. Nothing worked. The pathogen had redundancies for everything. Block one pathway and it switched to another. Catherine brought in Dr. Anthony Webb, a specialist in evolutionary biology. She showed him the genetic sequences without initially explaining their age. Anthony studied them for 3 days, running phonetic analyses, comparing them to known viral families. His conclusion was definitive. The sequences didn't fit any evolutionary pathway. This organism had features from multiple viral lineages combined in ways that don't occur through natural evolution. When Catherine revealed the samples were 75,000 years old, Anthony was stunned. Genetic engineering at this level didn't exist then. It barely exists now. Unless someone 75,000 years ago had knowledge we're only beginning to acquire. Catherine felt the implications settling over her. This wasn't a natural pathogen. It was a designed weapon, created by an ancient civilization with technology that shouldn't have existed. And it was now spreading through the modern world, erasing human memory with the same efficiency it must have used 75,000 years ago. Catherine's team worked around the clock to map exactly how the pathogen destroyed memories. Understanding the mechanism was crucial to finding a way to stop it. They used advanced imaging techniques to watch the process in real time. Fluorescent proteins marked the pathogen and its products, allowing visualization of infection spread through neural tissue. The pattern was consistent and terrifying. The pathogen initially infected neurons in the dentate gyrus, a specific region of the hippocampus where new memories first form. From there, it spread along neural pathways to other hippocampal regions. Each region plays a role in memory processing, and each became systematically infected. Once established, the pathogen began producing its memory-erasing proteins in massive quantities. The proteins had multiple effects. First, they interfered with glutamate signaling. Glutamate is the primary excitatory neurotransmitter in the brain, essential for neurons to communicate. The pathogen's proteins blocked glutamate receptors. Without proper signaling, neurons couldn't communicate effectively. Memories stored in patterns of neural communication began fading. Second, the proteins activated specialized enzymes called proteases that break down proteins. They targeted proteins that form the structural scaffold of synapses. As the proteases destroyed these structural proteins, synapses physically fell apart. The connection points between neurons disintegrated at the molecular level. Third, and perhaps most insidiously, the pathogen reprogrammed the neuron's own gene expression. It activated genes normally silenced in mature neurons, genes involved in pruning and eliminating connections. Simultaneously, it silenced genes responsible for maintaining and strengthening synapses. The result was neurons that actively destroyed their own connections while being unable to form new ones. The memory loss followed a specific temporal pattern. Recent memories disappeared first, followed by progressively older ones. This occurred because of how memories are stored in the brain. When you first form a memory, it's initially dependent on the hippocampus. The neural pattern is fragile, easily disrupted. Over time, through memory consolidation, the memory becomes encoded in connections throughout the cortex, independent of the hippocampus. The pathogen's initial infection of the hippocampus destroyed recent, unconsolidated memories immediately. But as it spread to other brain regions, it began attacking the cortical connections, storing older memories. The progression was predictable. Patients lost recent memories within days. Memories from weeks or months ago vanished within the first few weeks. Older memories survived longer but eventually succumbed. In late-stage patients, the memory loss was nearly complete. They couldn't remember their names, their families, their languages. They retained only the most fundamental abilities. But even those were beginning to degrade. Dr. Olivia Myers, the team's neuroscientist, made a crucial observation. She was examining brain scans of late-stage patients and noticed unusual activity in the prefrontal cortex and motor regions. The pathogen wasn't just destroying memories, it was rewiring neural pathways. Brain imaging showed new connections forming in regions normally associated with goal-directed behavior. But the connections were abnormal, creating circuits that didn't exist in healthy brains. Late-stage patients were no longer acting on their own will. The pathogen was controlling them. Catherine realized the true horror. This wasn't designed just to erase humanity's collective memory. It was designed to turn humans into weapons against themselves. While Catherine's team dissected the pathogen's mechanisms, epidemiologists worldwide tracked its spread, searching for patterns. Dr. James Morrison led the epidemiological analysis at the World Health Organization. He compiled case reports from every outbreak zone, mapping infection rates, transmission routes, demographic factors. What emerged was a pattern that shouldn't exist. The pathogen was spreading through water systems, but its spread didn't follow normal epidemiological models. Infection rates were too high. Transmission efficiency was too consistent. It infected almost everyone who encountered it. Immune systems didn't slow it. Genetic diversity didn't create resistant populations. It was as if the pathogen had been specifically designed to infect humans with near-perfect efficiency. James began mapping the outbreak's geographic distribution. Infections clustered around water sources connected to Arctic glacial systems, rivers fed by melting ice, coastal regions where Arctic currents flowed. But there was something else, a secondary pattern underlying the geographic spread. The outbreak was following routes that coincided with ancient human migration paths from 75,000 years ago, river valleys that early humans had traveled. Coastal areas where archaeological evidence suggested prehistoric settlements. It was as if the pathogen remembered the world as it had been during the last ice age. James shared his findings with Catherine. Together, they began reconstructing what might have happened 75,000 years ago. That time frame coincided with a genetic bottleneck in human evolution. DNA evidence shows human population crashed dramatically, like dropping to perhaps 10,000 individuals before slowly recovering. The traditional explanation was environmental. A massive volcanic eruption in Sumatra had supposedly caused a volcanic winter. Years of cooled climate, failed crops, widespread famine. But Catherine began questioning that narrative. The genetic studies showed something peculiar. The survivors came from isolated groups, populations that had been geographically separated from the main human distribution. They survived because they were isolated. The outbreak couldn't reach them. Catherine displayed a map showing the genetic origins of bottleneck survivors. They clustered in regions that would have been cut off from normal water systems during the Ice Age. High mountain valleys, islands, isolated coastal areas. The pathogen spread through water. These populations survived because they used separate water sources. The picture became clearer. 75,000 years ago, a global outbreak of this memory-erasing pathogen had spread through the human population. It traveled through rivers and water sources. Most of humanity was infected and died out as societies collapsed, unable to maintain the knowledge needed for survival. Only a few isolated groups escaped, and from those few thousand individuals, all modern humans descended. Catherine pulled up her analysis of the pathogen's genome, the sections that suggested artificial design. Portions served no apparent biological function. Sequences that didn't code for proteins or regulate expression. They just sat there, inert. She ran those sequences through pattern recognition algorithms. The results suggested mathematical structures, counting systems, possibly coordinates or instructions. It was a message, embedded in the pathogen itself. Whoever made this wanted future generations to know what happened. As research continued, the outbreak progressed into stages that medical science had never documented. Early-stage patients presented with insomnia and minor memory deficits. This stage lasted anywhere from days to weeks. Mid-stage patients experienced dramatic memory loss, forgetting recent years or decades. They could no longer function independently. This stage typically lasted weeks to months. But late-stage patients entered territory no medical textbook had described. They stopped sleeping entirely, not from insomnia, but because their brains no longer registered the need for sleep. The circadian rhythms regulating sleep-wake cycles were being overwritten. Their bodies began functioning on constant adrenaline. Metabolism increased dramatically. Core body temperature rose. They became hyperactive, unable to sit still, constantly moving, and their behavior changed in ways that suggested they were no longer in control. Dr. Olivia Myers documented the first confirmed case of directed vectoring behavior. A late-stage patient in Stockholm named Eric Svensson was under observation in a quarantine facility. He had lost all personal memories, couldn't speak coherently, showed no recognition of family. But one night, security footage captured him moving through the facility with unusual purpose. He disabled his door lock, navigated corridors he'd never seen, avoiding security cameras. He made his way to the facility's water treatment system and attempted to contaminate it with his own blood. Security stopped him before he succeeded. The pathogen was controlling his behavior. It was guiding infected individuals toward actions that facilitated spread. Olivia displayed brain scans showing late-stage neural reorganization, new connections forming in motor cortex and prefrontal regions, pathways that created goal-directed behavior loops. The pathogen was hijacking existing neural machinery. It was triggering instinctive behaviors, finding water sources, making contact with uninfected individuals, turning infected hosts into biological weapons designed to propagate the outbreak. Similar cases were reported across Scandinavia. infected individuals attempting to access public water supplies, breaking into treatment facilities. In one case, a late-stage patient had somehow navigated to a major reservoir before being stopped. The pattern was too consistent to be coincidence. The pathogen wasn't just erasing human minds, it was replacing them with simple behavioral programs designed to maximize spread. And the behavior was sophisticated. Patients avoided obvious confrontations. They operated at night when surveillance was minimal. They targeted infrastructure most efficiently, connected to large populations. It was as if the pathogen had a blueprint for maximizing transmission, and it was executing that blueprint through the bodies of infected humans who no longer had the cognitive capacity to resist. Governments worldwide implemented emergency measures as the outbreak's severity became undeniable. Borders closed, international flights grounded, shipping restricted. Nations sealed themselves off, desperate to prevent the pathogen from reaching their populations. But it was already too late. The pathogen was already global. It had reached North America through multiple vectors: cargo ships with infected crew, air travelers incubating the pathogen, ocean currents carrying viral particles to coastal water sources. The eastern seaboard reported cases: Boston, New York, Washington. The pattern was identical to Scandinavia: insomnia, memory loss, progressive deterioration. The Gulf Coast followed, then the Pacific Northwest, then inland regions as rivers carried the pathogen into America's heartland. Asia saw outbreaks following similar patterns. Japan's extensive coastline made it vulnerable. China reported cases in the north, then spreading south. India saw infections in coastal communities before the pathogen moved inland. Europe was already overwhelmed. The Rhine carried it from the Alps to the North Sea. The Danube brought it through Central Europe. The Thames, the Seine, the Po all became conduits. Africa reported outbreaks in the north. South America saw cases in countries with connections to Atlantic shipping routes. The pandemic everyone had feared was here, and it was worse than imagined because you couldn't see it coming. No fever, no cough, no visible symptoms until memory loss began. By then, patients had been infectious for weeks. Hospitals became overwhelmed with individuals who needed total care. They had lost the ability to function independently. They forgot how to eat, how to dress, eventually how to recognize other humans. Healthcare systems buckled. Staff-to-patient ratios became impossibly inadequate. Infrastructure began breaking down. Water treatment facilities couldn't operate when engineers forgot their training. Power grids failed. Supply chains collapsed. Society wasn't collapsing from violence, it was dissolving from collective amnesia. The accumulated knowledge that kept civilization functioning was being erased. Some nations implemented radical quarantine measures. Island nations hoped geographic isolation would protect them, but the pathogen was already in the water. Ocean currents didn't respect quarantine. Viral particles carried by currents reached remote islands. Reports emerged of infected populations being abandoned, entire cities sealed without resources. Mass graves where late-stage patients were disposed of. No ethical solution existed. The pathogen was too infectious, too resilient, too widespread. Traditional public health measures were useless. And through it all, Catherine's laboratory in Washington worked frantically. Catherine had barely slept in weeks. Her team cycled through potential treatments, testing compounds, analyzing failures, adjusting approaches. They had tried every antiviral in existence. Nothing worked. The pathogen had resistance mechanisms for everything. They attempted immunological approaches, vaccines, antibodies, T-cell therapies. All failed. The pathogen evaded immune recognition. They tested neuroprotective compounds. The pathogen adapted, upregulating its synaptic-destroying proteins. Every avenue seemed blocked. But then Catherine noticed something unusual in the pathogen's genome that she had previously dismissed. Scattered throughout the genetic sequence were regulatory elements that appeared designed to shut down viral replication under specific conditions. Kill switches. Multiple redundant systems. Why would a weapon have off switches? She studied these sequences obsessively. They required specific molecular triggers, proteins that didn't exist in modern biology, compounds that would need to be synthesized from scratch. One sequence caught her attention. It responded to a protein with a structure she recognized from her ice core research, a molecular signature found in ice layers, deposited immediately after the outbreak 75,000 years ago. As if whoever created the pathogen had also created the cure and frozen it in the glaciers. The realization hit Catherine like electricity. This wasn't just a weapon, it was a test or a message. The ancient civilization had left instructions for stopping it, encoded in the same ice that preserved the threat. But synthesizing the cure protein was monumentally complex. The chemical synthesis would take months they didn't have, unless the protein itself had been preserved in the ice. Catherine pulled up her archives. She needed ice cores from precisely dated layers, ice that had frozen between 74 and 73,000 years ago. She sent urgent requests to research stations. The samples arrived within days. Catherine's team worked around the clock extracting and analyzing the ancient ice. And there, suspended in frozen water 75,000 years old, they found it. The cure protein, perfectly preserved, still functional after millennia. Catherine immediately began scaling up extraction protocols, but even working at maximum capacity, they could only extract tiny amounts, not nearly enough. They needed to manufacture it. The team used advanced crystallography to map the protein's three-dimensional architecture. As they analyzed its structure, they began understanding how it worked. The cure protein targeted the kill switches embedded in the pathogen's genome. It bound to specific viral proteins and triggered a cascade that shut down viral replication permanently. But more than that, it activated cellular repair mechanisms. It stimulated production of proteins that could rebuild damaged synapses. It promoted neurogenesis. It wasn't just stopping the pathogen, it was repairing the damage. Catherine authorized clinical trials. Late-stage patients were given experimental doses extracted from ice cores. The results were dramatic. Within hours, viral load in cerebrospinal fluid began dropping. Within days, brain scans showed reduced inflammation. Damaged neural tissue showed signs of repair, but the memories didn't come back. That was the heartbreaking reality. The cure could stop further damage and promote repair, but it couldn't restore synaptic connections that had been physically destroyed. The neural networks storing memories were gone. Treated patients survived, but they were empty vessels. They had to relearn everything from scratch. It was better than continuing to deteriorate. But it wasn't the miracle cure Catherine had hoped for. Still, it was something. A way to halt the outbreak, a way to save at least some cognitive function if treatment could be administered early. Now they just needed to manufacture it on a global scale. While pharmaceutical companies mobilized to produce the cure protein, Catherine continued studying the markers embedded in the pathogen's genome. Dr. Lisa Park, a mathematician on the team, noticed that the sequences contained prime number patterns. Not random primes, but specific sequences that appeared to encode coordinates. They weren't just numbers. They were directions, latitude and longitude. They plotted the coordinates on maps of current Arctic geography. Each point corresponded to a location where significant glacial melting had occurred. Rock formations and cave systems that had been buried under ice for millennia were now exposed. Catherine organized expeditions to these sites. Research teams found carved messages, not written in language but in mathematical notation and symbolic diagrams. The carvings told a story that changed everything. 75,000 years ago, human cognitive evolution had reached a tipping point. One population had developed significantly advanced technology. They understood genetics, molecular biology, neuroscience at levels modern science was only beginning to match. But they were not unified. Multiple factions existed with radically different philosophies. One faction believed that advanced knowledge was dangerous, that accumulated wisdom led to violence and cycles of destruction. They advocated for periodic resets, erasing collective memory to give civilization fresh starts. They created the pathogen as a tool for this reset. The carved messages showed diagrams of the pathogen's structure, its mechanisms, its purpose. It was designed to spread globally, to erase human memory while leaving basic survival instincts intact. But they hadn't fully understood what they were creating. They thought they were erasing knowledge. Instead, they were erasing the capacity to accumulate knowledge. The pathogen didn't just delete memories, it damaged the neural structures needed to form new ones. Their experiment went catastrophically wrong. The pathogen spread faster than anticipated. It mutated, becoming more aggressive. Within years, it had infected most of the global population. Civilizations collapsed, not from death, but from forgetting. The infrastructure crumbled. People regressed to primitive survival. Most died. The opposing faction recognized the catastrophe. They dedicated their final resources to creating a cure and preserving knowledge. They engineered the cure protein and froze it in glaciers. They carved warnings in stone and buried them under ice. They left markers in the pathogen's genome, hoping a future species would decode the message. Then they died, either from the outbreak or from civilization's collapse. The carved messages ended with a warning. We sought to remake ourselves by erasing the past. We believed knowledge was corruption, that memory was burden. We were wrong. Memory is not the disease, it is the foundation. Without it, we are not human. Without it, civilization cannot exist. Learn from our error. Do not repeat our mistake. The ice preserved our failure. Respect that preservation. What sleeps beneath should remain frozen. We are the cautionary tale. Do not become the next. Manufacturing the cure protein became a global undertaking. Pharmaceutical companies collaborated in unprecedented ways. Patent protections suspended, trade secrets shared, production facilities worldwide retooled. But production couldn't keep pace with demand. Billions needed treatment. Manufacturing capacity could only produce enough for millions. Distribution became a nightmare of ethical calculations. Who received treatment first? Early-stage patients who could recover most function? Healthcare workers essential for infrastructure? Leaders necessary for organizing response? Every choice meant condemning others to deterioration. Some nations hoarded production. Others attempted fair global distribution. Black markets emerged where the wealthy purchased treatment while the poor went without. The treatment itself required careful administration, direct injection into cerebrospinal fluid. This meant trained medical professionals, sterile facilities, time-consuming procedures, all scarce resources. And treatment only worked if administered before too much neural damage occurred. Early-stage patients could retain most memories. Mid-stage patients recovered partial function. Late-stage patients benefited least. The tragic irony was that late-stage patients needed intervention most urgently but gained the least benefit. Still, stopping viral replication in even late-stage patients prevented the directed vectoring behavior. It halted the systematic spreading of infection. Catherine coordinated with the WHO to optimize distribution strategies. They developed triage protocols, prioritized populations, implemented mobile treatment units. It was brutal calculus. Every protocol meant explicitly deciding that some people would not receive treatment in time. The cure rolled out over months. Production gradually increased. Distribution networks stabilized, infection rates began declining, but the damage was immense and permanent. The pandemic burned through human populations for 3 years before treatment became widespread enough to control it. The final numbers were catastrophic. Roughly a third of global population had been infected at stages too advanced for meaningful recovery. They survived treatment, but they had lost everything. Another 40% had experienced early or mid-stage infection. They retained some memories, but with significant gaps. Skills degraded, knowledge fragmented. Only about a quarter of humanity emerged relatively intact. Civilization didn't collapse completely, but it was fundamentally transformed. Nations that had been technological powers struggled with basics. Entire fields of specialized knowledge had been lost when experts forgot their expertise. Libraries and digital archives survived, but having information isn't the same as having people who understand it. Rebuilding became the primary focus. Retraining infected individuals, teaching them to read again, to calculate, to function. It was like raising a generation of adults from childhood. Some adapted better than others. Younger patients relearned faster, but older individuals struggled. Many never fully recovered. The psychological impact was equally devastating. Survivors grieved for their lost selves. They had memories of being competent, skilled, knowledgeable. But those abilities were gone, relationships shattered. Spouses cared for partners who no longer remembered their shared history. Parents forgot their children. The social fabric had been torn apart by mass amnesia. Catherine continued her research, now focused on understanding why the ancient civilization had created such a weapon and left instructions for stopping it. One passage kept drawing her attention from the carved messages. The faction that created the pathogen believed accumulated knowledge led to rigid thinking. That civilization's problems stemmed from being trapped by history. They thought erasing memory would free humanity. Catherine understood the appeal of that philosophy, but the ancient experiment had proven the fatal flaw. Memory isn't just historical record, it's the foundation of learning itself. Without the ability to remember, you can't improve, can't adapt, can't build on past successes or learn from failures. Erasing memory doesn't create freedom, it creates helplessness. The ancient civilization learned that lesson too late. The survivors had to rebuild from scratch. It took them tens of thousands of years to redevelop technology, and they never recovered the heights their ancestors had reached. Modern humanity was following the same trajectory. The outbreak had erased so much accumulated knowledge that it would take generations to recover. The carved messages ended with coordinates to one final location. A site in the Canadian Arctic recently exposed by melting ice. Catherine organized an expedition there. The site in the Canadian Arctic was a cavern carved deep into rock that had been buried under ice for 75,000 years. Inside, the ancient civilization had left their final message, preserved in synthetic polymers that encoded information at the molecular level. Technology that shouldn't have existed millennia ago. Catherine's team extracted the polymers and decoded their contents. What emerged was a complete archive: history, science, philosophy. The accumulated knowledge of the civilization that had created the pathogen. They had been more advanced than Catherine imagined. They had mastered genetic engineering, nanotechnology, artificial intelligence. They had explored space. They had extended human lifespan. They had achieved things modern science could barely conceptualize. But they had also been deeply divided. The archive contained arguments between factions. One insisted advanced knowledge bred arrogance, that civilization needed periodic resets. The other argued knowledge was humanity's only defense, that erasing it was suicide. Eventually, one faction gained enough power to implement their solution. They created the pathogen, released it, believing they could control the outcome. They were catastrophically wrong. The archive showed the collapse in detail: cities abandoned, fields left unplanted, mass death from starvation, disease, violence as social structures dissolved. The faction watching their experiment spiral out of control realized they had destroyed their own civilization. They created the cure too late. By the time it was distributed, most of humanity was gone. Advanced civilization was over. The survivors recorded their story as a warning. They left the cure in ice. They carved instructions in stone. They buried their knowledge, hoping someday a future civilization would discover it and learn from their mistake. The archive's final entry was addressed to whoever eventually found it. We believed we could improve humanity by erasing its memory. We thought knowledge was corruption. We were arrogant. We were wrong. Memory is not humanity's flaw. It is our essence. Without it, we cannot learn, cannot grow, cannot pass wisdom between generations. We destroyed our civilization trying to improve it. Do not make our mistake. Respect memory, respect history, respect the accumulated knowledge of your ancestors. It is flawed, it is incomplete, but it is all that stands between civilization and oblivion. The glaciers preserved our failure as a warning. Now those glaciers are melting. Other dangers sleep in the ice, pathogens we created and could not control. Technologies we abandoned as too dangerous, experiments that failed catastrophically. They are waking as the ice melts. You have survived one. There are more. The ice is not your enemy, it is your guardian. What we froze should remain frozen. What you have awakened once must never be awakened again. Learn from us. We reached heights you have not yet achieved. And we fell because we forgot that memory is sacred. Do not repeat our error. Honor your history. Preserve your knowledge. Respect the past. It is the only foundation for the future. Catherine returned from the Arctic expedition changed. She had gone searching for answers. She had found a warning that terrified her more than the outbreak itself. Climate change was still accelerating. Glaciers were melting faster every year. And if one ancient pathogen had been released, what else was waiting? She dedicated the remainder of her career to a new mission, identifying other potential threats before they thawed into the modern world. She established the Glacial Monitoring Initiative, a global network studying ice cores, mapping ancient biological materials, creating early warning systems. The work was overwhelming. Millions of square kilometers of ice, countless ancient layers, each potentially harboring preserved pathogens, toxins, or technologies from civilizations that had failed millennia ago. Catherine's team found evidence of other engineered organisms in ice cores. Some appeared to be failed experiments. Others looked like weapons never deployed. A few were so alien in design that understanding their purpose seemed impossible. Each discovery reinforced the message. The ice was a tomb, and humanity was digging it up without understanding what it had buried. 5 years after the outbreak was contained, Catherine gave her final lecture before retiring. The auditorium at Washington University was filled with students, colleagues, and policymakers. Her message wasn't triumphant, it was terrifying. We think of ourselves as advanced, as prepared. We developed the cure that stopped the outbreak. We rebuilt our infrastructure. We implemented protocols for future pandemics, and we learned nothing. The glaciers are still melting, faster now than when this began. We're still releasing whatever the ancient world locked away. We survived one outbreak through luck and the mercy of a dead civilization that left us instructions. We might not be lucky again. There are other things in the ice, other pathogens, other technologies, other experiments that failed catastrophically. They're waking up as we speak. The ice cores show evidence of at least 17 distinct engineered organisms preserved from the last glacial period. We've encountered one. 16 more are melting into the water supply right now, and those are just the ones we can identify. The ancient civilization that created this pathogen was more advanced than us, and they died. They collapsed because they made a mistake they couldn't fix. The ice preserved their mistake as a warning. Now we're melting that ice, releasing everything they locked away, and we're doing it deliberately. We call it climate change. We treat it as an environmental problem. But it's archaeological. We're excavating a mass grave of failed civilizations without protective equipment. The pathogen we survived was meant to erase human memory. What if the next one erases something else? The ice holds answers, and we're melting it as fast as we can, convinced we're too advanced to face consequences. We're not. The last civilization thought that too. They created a weapon they couldn't control. It destroyed them. The ice kept it frozen for 75,000 years. We melted it in a few decades. What else are we melting? What other mistakes are we about to repeat? The auditorium was silent. The message from the ancient civilization was clear. Respect the ice, respect what it preserved, respect the warnings it contains. We didn't listen. We're still not listening. Climate change is real. Its effects are catastrophic. But the danger isn't just rising seas and extreme weather, it's what comes out of the melting ice. We need to be studying those glaciers, mapping what they contain, preparing for what might emerge, not waiting for the next outbreak to surprise us. The ice was our guardian. We're destroying it, and when it's gone, we'll face everything it was protecting us from. All at once, without preparation, without understanding. That's the real threat, not the pathogen we survived, but the ones we haven't encountered yet. They're coming. The ice is melting, and we're not ready. The glaciers are still melting, faster now than ever before. Every year, rivers swell with ancient water, water that has been frozen since before human civilization began, water that carries microscopic passengers from a world that died millennia ago. Most are harmless, some are not. The Ice Age didn't end because the climate warmed. It ended because something killed so many humans that the survivors couldn't maintain their technology, couldn't keep their knowledge alive. We nearly went extinct once before. The ice preserved that truth, locked away the weapons that caused it, protected us from the mistakes of civilizations that failed long before ours began. And now we're melting it away, one glacier at a time, releasing everything the ancient world tried to bury, convinced that we're advanced enough, intelligent enough, prepared enough to handle whatever emerges. We're not. The pathogen that almost erased humanity was just the first warning. There are more coming, thawing in the ice right now, waiting to remind us that progress isn't protection, that technology isn't wisdom, that every civilization, no matter how advanced, is just one mistake away from extinction. The ice remembers, even if we don't.